Hacer una tesis doctoral en ciencias experimentales es un reto vital. Durante ese periodo, los experimentos pueden salir mal, tu proyecto científico puede fracasar, puedes encontrarte con un ambiente de trabajo hostil, tienes competidores, pero quizá la realización de una tesis doctoral sea uno de los mejores ejemplos del aforismo que dice que lo importante no es la meta, sino el camino. Durante la tesis conoces gente con personalidades muy variadas y de culturas muy distintas. Aprendes a colaborar con ellos, entiendes la importancia del rigor científico, de plantearte las preguntas de manera adecuada, aprendes a sobreponerte a la frustración de un experimento fallido o de un artículo científico rechazado. En definitiva, es un trayecto vital pleno de experiencias que te ayudan a madurar como persona y como científico, independientemente de que tus investigaciones hayan dado resultados más o menos relevantes. Por eso yo suelo decir a los jóvenes investigadores que al acabar una tesis ya han ganado, y que se olviden de lo que la sociedad o el entorno reconoce como éxito, que podría ser una publicación impactante o salir en prensa o en medios de comunicación. Les digo que lo importante durante ese periodo, y en todos los periodos de la vida en general, no es tanto el supuesto éxito como la satisfacción de haber superado retos y de haber crecido como persona y como científico. Y si ninguno de tus experimentos ha funcionado, acuérdate de la frase de Thomas Alva Edison que dijo: «Yo no he fracasado, he encontrado 10.000 maneras que no funcionan». Este episodio de Raticos de Ciencia es un episodio, digamos, experimental. Los investigadores predoctorales del IBEC, Instituto de Bioingeniería de Cataluña, celebraron su PhD Day, o Día de los Investigadores Predoctorales, y me invitaron a hablarles sobre cómo hacer un podcast de ciencia. Y decidimos convertirlo en un taller en el que se entrevistarían entre ellos. Aquí escucharéis a 14 grupos de 2 personas haciendo preguntas a una tercera, de su investigación, de su vida personal y de sus intereses más allá de la ciencia, siguiendo un poco la línea de Raticos de Ciencia de no separar ciencia de humanismo. Lo hicieron según su conveniencia en inglés, castellano y catalán. Aquel día me encontré a jóvenes investigadores de distintas nacionalidades, llenos de energía y de ilusión, brillantes y abiertos de mente. Ojalá la ciencia se porte bien con ellos y mantengan el entusiasmo que percibí, porque, como dijo Winston Churchill, "Success is going from failure to failure without losing enthusiasm". El èxit és anar de fracàs en fracàs sense perdre l'entusiasme. El éxito es ir de fracaso en fracaso sin perder el entusiasmo.
Raticos de Ciencia. Conversaciones con científicos y científicas. La ciencia, como la vida, se disfruta a raticos.
We are in a beautiful place, in a rainy day at the beginning, now sunny, close to the coast in the south of Barcelona. We are in a Casa de Colonias in Spanish. In English, we don't know how to translate this, but it's kind of a beautiful house with bed and with this kind of cheap to visit for a big group. And the group of people that is here today are PhD students of the Institute of Biotecnología. How is the name?
Institute of Bioengineering of Cataluña.
Exactly. I didn't prepare well this. Thank you guys for inviting me today to talk about how I'm doing science communication. A podcast was really nice. I really feel for the very beginning a lot of energy in the audience because everyone is a PhD student from many different places. And I have to say thank you to you for inviting me, but also for being brave of organizing this, because I think we all depend on people that have the courage of organize stuff. We need people like you to make things happen.
Thank you.
Well, we're going to start with this, Gala. So how did you end up organizing this event?
I think that we kind of started with Janet together, joining the PhD committee at the same time. And it's because, we like doing research. We like doing also outreach activities. But we think that it's really important to engage with your colleagues and try to know each other better. And usually with our kind of busy schedules, we don't have much time to get to know each other. And then, like for, I don't know how many years, we have been doing the retreat in IBEC.
I think this is the 6th year, something like this.
Okay.
Yeah. So there has been some people before us, also in the PhD committee, organizing this. So the real pioneers were the first ones doing this. Now repeating it every year, we already have some ground before us.
Okay.
So.
What is your main motivation to do this? Is like to know people, to really make life easy to people, or because you like, let's name it, you like the mambo, the organized stuff?
A bit of everything. I like to make things happen, let's say. I think I'm a very active person, and I like organizing activities, meeting new people while you're organizing, but also create a space where people can meet others in organizing that. And I think it's also in my character to do that.
Okay. I think one of the most beautiful things about this retreat is because your institute is very international. I mean, you can find people from many different places. So tell me a little bit, Gal, about this, about how international is your institute and how do you feel working in a place with people with many different cultures?
I think that in IBEC, we are almost 50-50 people from Spain and 50% people from outside Spain. Also, we are really happy this year because we had a huge representation of the different groups of IBEC, although we are still wanting more representation from other groups. But I think that it's really interesting to have this chance to talk with people from different places from the world, and talking about science, but also outside science, because I think that it's also really important what you do with your podcast. It's showing that scientists, maybe they have a little bit of intelligence, to say it somehow, but we are probably not just brains. We also enjoy doing other activities, and we also have hobbies like normal people, I would say. So,
Yeah. So, Janet, can you tell me a little bit about the program for these 2 days? Because maybe for people that is outside science doesn't know what is this about, why to make a PhD retreat, and how do you organize 1 day and a half of activities?
Yeah, so we have been rotating different topics. So we've gone through career prospects. Last year it was more about mental health and well-being. And this year we thought that science communication would be a great topic because we're all curious to know more how to communicate our science, how to do outreach, because we're there stuck in the lab doing our experiments. But in the end, we need to explain to our families, to our friends, to everyone out there what is it what we are doing, And everyone's like, oh, no, I don't understand what you're doing. So trying to find our own way to explain this science. And this is why these 2 days are going to be about science communication in many different forms. So we started this morning with a podcast. Now in the afternoon, it's going to be more about how to do the outreach, or how to find the language to translate our... So it could be kind of a translation, right? Not using the scientific terms and using more terms that are more close to the general public. Then we're also going to do one more about art. Because,
interesting.
Yes, in art, you can also express something in a very different way. Maybe with an artistic view, you could see something in your data, maybe. Not just showing it always how we are doing it, but also in any paper that you write, there's going to be figures, etc. So art is very present in our lives. And one more that is more how to present, because as scientists, we need to present in front of an audience. And sometimes we are shy, we don't know how to do it. So this is also...
As one of my mentors says, it's important to do science, really cool science. But if you are not able to show what you're doing, then it's really important to work on that point as well as science.
And also I agree that art is important. I'm really happy to see that in the program, because I think at the end, most of the people doing science are very creative, and they are doing something beside science that has to do with creativity. And creativity, you can train that in many different ways. So what are you doing for your creativity outside of the lab?
Oh, wow, that's a good question. So I think one of the things I like to do is theater. So that would be my most creative things to do. But I also like sewing, painting, a bit of arts and crafts, let's say. I mean, on that side.
And the theater, you are going like every week to do this? Or?
In my case, it's more by project. It's an amateur group. So whenever there's one that wrote like a piece or made a project, and then we meet every week.
Okay.
To represent it.
So is any of this representation is available in YouTube or something like this?
No, I'm afraid it's a very it's you have to live it in the moment.
Okay.
It's a very kind of a fieldy experience.
Yeah, it's true. Like the theater is something just to live it in the moment. So what about you, Yara?
I think that in my case, I use creativity to play the drums. I play in the percussion section of my band in Cerdanyola del Vallès. And there you learn how to play in teams, no? Because each one has a different role. The triangle, although a lot of people think that it may be simple, you have to count, for example, 200 bars and then place the note in the place that it has to be. But you need creativity some way, like sometimes to be able to play two instruments at the same time and all that stuff. So I think that this is something that really energizes me outside of work.
Wow. So you can play like a whole song with a triangle, and then people recognize that this is whatever song you are playing? Or you need something else, or this mod an instrument to be next to other instrument?
Usually, in my case, what I do is that I play the timpanis, the snare drum, and the xylophone at the same time.
Wow.
So then I have to switch between them depending on the song.
Very interesting. Okay, so one last question because we have to move to the next session. So I would like to ask you, because you are like young scientist with I will say a lot of energy. You are a very nice institute doing a PhD. So if you can make or build a dream about how will be your job in 10 years, how will be that job? Will be a job in science, out of science, in industry, teaching? So dream a little bit. So tell me.
Can I say something? I think that I was told since I started the PhD that this is the forbidden question. So please,
okay, I like forbidden.
That being said, please stop.
I like the forbidden thing, but this is a dream. This is just a pure speculation. How do you envision yourself in 10 years? How would you like to be working in something related with science in 10 years?
This is a very, very tough question.
I mean, you don't have to develop, but something simple. I mean, first question is, you see yourself in science in 10 years?
I think I could see myself in science, yes. And I could see myself also not in science. Which is a very broad answer, but I think this is also inherent in a scientist's curiosity. Because I really like science about how also for curiosity and like search of knowledge, but also how it serves society as well. No, if you go more translational. So it could also be another.
You guys are smart people, and this is a very smart choice. I had a plan B that is okay, because I think when people focus in a plan A, put all the energy in a plan A, could lead a lot of frustration. Today, in the first slide of my talk, I was showing this slide of Groucho Marx. He have a sentence saying that these are my principles. If you don't like it, I have another one. So it's always to have a plan B, a different one that you feel comfortable with. So I like that. So what about you, Dara?
I think that in my case, I hope, or at least I will try to work to be in a position that I really enjoy going there every day or doing it every day. So something that really, it's my passion, I would say. And yeah, I think that I agree with Janet. I don't think that that I belong to academia or to industry. I don't know. I think that I will take whatever it comes and how it comes, and yeah, we will see.
Very nice. Very is also a smart answer. I think to find a place where the Monday morning arrive and it's not a drama, it's kind of a joy. So this is, at the end, this is success, right? So good luck to both of you.
Thank you.
Thank you again for your effort to put all this together. I have one last question. I see in the program that you have a custom party and then a crazy dinner. What means crazy dinner? It's a pity I'm not going to be here, but I'm curious about this crazy dinner.
If you can explain, Gala.
Well, the costume party this time the topic is about masterpiece. So we are art masterpiece. So yeah, we will see what we find in the end of the day. And the crazy dinner. I think that we started this last year. And it's basically you have different prompts, and you have to do that prompt. For example, I don't know, start shouting or just clap your
singing
hands.
a song whenever someone has gone and given a hug to somebody else. So it's a chain of activities that are going to happen during dinner to make it more fun.
Okay, interesting. I think that there is sangria around. I think you are gonna need a bit of sangria before.
But IBEC doesn't need to know.
Okay. It's sangria free of alcohol, of course.
Yes. No ethanol.
Okay. Thank you very much for your time and have fun.
I think that we also need to thank you for coming here, for giving us this workshop on how to make a podcast. Has been super interesting. The feedback from everyone was that they enjoyed very much participating and engaging a lot in the activity, and maybe we'll see some podcasts coming from this.
Sure. Thank you.
Thank you very much.
Bueno, Ricard, tú nos estabas explicando en qué consiste un poco tu proyecto de la tesis doctoral y nos has comentado que estudias ciencia básica en la biología del desarrollo.
Así es.
Y bueno, nos gustaría saber un poco más en qué consiste tu investigación. Cuéntame qué es exactamente lo que estudias y cómo lo haces.
Perfecto. Pues nosotros usamos modelos muy simples de gastrulación humana porque realmente mirar dentro del embrión es o muy complicado o imposible en el caso del humano, ya que no tenemos derecho a cultivar embriones humanos por más de 14 días. Entonces lo que hacemos es poner células que diferenciamos en un entorno en el que nosotros creemos que es parecido a la fisiología normal. Tenemos células que se organizan por sí mismas en las 3 capas del embrión real. Y lo que nosotros queremos hacer es estudiar su comportamiento y relacionar los diferentes estadios de diferenciación con diferentes comportamientos desde un punto de vista mecanobiológico. También la pregunta que nos hacemos de forma principal es si realmente hay un rol entre mecánica, fuerzas, tracciones y la diferenciación de estas células. Es un poco la pregunta del huevo y la gallina.
Uh-huh.
¿Son estos cambios en el entorno y de comunicación entre células las que causan que las diferentes células se diferencien más rápido, menos rápido, se muevan, no se muevan tanto, hacia dónde? O si, por otra parte, estos cambios en las tracciones de estas células, las fuerzas que ejercen estas células, se deben a diferentes estadios de diferenciación. Para que os hagáis un poco mejor la idea, tenemos células que son extraembriónicas, que son las que en un contexto real formarían la placenta. Tenemos células que son amnióticas, que son las que formarán el saco amniótico del embrión. Y luego tenemos células de mesodermo, que son células que pasan de epiteliales, es decir, de una capa más inmóvil, a mesenquimales, que quieren migrar hacia donde quieren. Y luego tenemos células de endodermo, que son las células que realmente formarán los órganos internos. Las células que se mueven más respecto a las células que se mueven menos van a tener diferentes patrones de fuerza en unos geles donde las sembramos. Con suficiente resolución temporal y espacial podemos empezar a preguntarnos realmente más en profundidad si estos cambios que nosotros leemos vienen antes o después de diferenciación de las células o si son formas perfectamente plausibles de diferenciar estas células solamente en función de su comportamiento mecánico.
Porque volviendo un poco a lo que has dicho antes de la pregunta de qué fue antes el huevo o la gallina, ¿vosotros utilizáis algún tipo de tratamiento con citoquinas, factores, para inducir la diferenciación o es espontáneo? O, por otra parte, ¿aplicáis algún tipo de fuerza o movimiento que pueda acabar, pues como tú dices, induciendo la diferenciación de estas células o no? ¿Es espontáneo?
Pues de momento nosotros usamos un modelo que se basa en añadir un morfógeno y eso hace que las células se diferencien por sí solas, pero la intención a largo plazo es causar la diferenciación de estas células mediante perturbaciones de carácter puramente mecánico. Es decir, si yo cojo mis colonias de células y las estiro o las comprimo, que son dos perturbaciones que las células sienten de forma muy normal en muchos contextos fisiológicos, quizás estos estímulos son suficientes o necesarios para que estas células se diferencien en ausencia de morfógenos externos.
Bueno, son preguntas muy, muy curiosas, la verdad, las que te planteas en tu doctorado. Y bueno, creo que nos pueden dar respuesta a un proceso tan interesante como es el desarrollo embrionario, del que se conoce tan poco, sobre todo por el difícil acceso, ¿no? Y bueno, aparte del laboratorio, ¿qué otras curiosidades tienes en tu vida? ¿Qué hobbies tienes?
Pues al trabajar en desarrollo humano tengo horarios bastante complicados porque soy un poco esclavo de lo que hacen mis células, pero cuando puedo intento ir al cine. Me gusta mucho el cine de todos los tipos y tengo un grupo de amigos desde muy chiquito con el cual tengo un equipo de fútbol y entrenamos y jugamos una vez a la semana muy tarde en Cerdanyola del Vallès y eso es básicamente lo que hago con mi tiempo libre.
Qué guay. ¿Y dirías que hay algo en común entre tus hobbies, alguno de tus hobbies como el cine o el fútbol con la ciencia o con tu proyecto?
Realmente a veces para explicarles a mis amigos un poco lo que hago en mi trabajo, porque muchos de ellos no tienen formación científica, pues intento usar analogías entre las diferentes posiciones que toman los jugadores en el campo y estas células mías. Pues tenemos algunas que actúan más como barreras, ¿no? Serían las células amnióticas, que son células más planas, que restringen más el movimiento. Eso lo podríamos relacionar más con la tarea que haría un defensa. Luego tenemos las células del mesodermo, que se mueven mucho y muy rápido, algunas de ellas, y van un poco por su cuenta. Serían ese tipo de jugador más creativo, quizás, o que va por banda y regatea más. Y luego tenemos células del endodermo, que son las que están en el centro de mis colonias, que tampoco se mueven mucho, pero son de las que menos sabemos realmente, porque de momento no hemos encontrado ninguna relación entre estas fuerzas que ellas ejercen y su comportamiento, pero sabemos que son importantes a nivel de señalización y quizás estas células las podríamos relacionar más con los jugadores del medio del campo, que son las que organizan un poco al resto. Y eso es un poco lo que intento explicarles a mis amigos.
Yo creo que es una buena forma de acercar la gente a todo el campo científico, ¿no? Bueno, pues muchas gracias, Ricard. Te deseo mucha suerte en tu proyecto.
Sí. Hasta la próxima.
Adiós.
Good morning. We are nanoproofs and nanoswitches PhD students. I'm Marina.
I'm Nayeli.
And we're working at IBEC. Today, we are interviewing Tom.
Hi, Tom. How are you?
I'm very good. It's a pleasure to be here.
Oh.
Thank you for hosting.
Yeah, it's a little bit hard to maintain the composture, to try to be very calm in this podcast. So we are trying to do the best. But we want to start to ask you about how it's going the PhD, your project. You will work in this kind of physicists, chemistry things, You can explain a little bit about what is going in your PhD?
Also, we know that you obtain a fellowship.
Ah, that's true. Congratulations.
Thank you so much. Thank you so much. I'm on it.
Yeah. Perfect.
So to begin, I'm just starting my PhD now, about to start my first year.
Perfect.
And it's the beginning of a long journey.
Yeah, it will be a long journey.
A long journey. So it feels strange now to talk about this already, but I can maybe tell you a bit about my project.
Okay.
So I work, as well as the IBEC, I work with the Hospital San Juan de Deo.
Oh, so cool.
And there, the lab specializes in a type of disease known as collagen 6 muscular dystrophy.
Okay.
And this is actually a really rare type of disease, which affects probably 1 in 100,000 people. So it's really rare. And so it's quite exciting to be involved in a project where not much is known, or at least the knowledge is continuing to be developed. And for this disease, patients unfortunately can't produce a specific type of collagen. So we all know collagen in skincare and this kind of thing. It's a type of protein which is actually everywhere in the body. And for these patients, unfortunately, because this protein is missing, they have muscle weakness, they're unable to walk, they lose the ability to breathe at some stage, and unfortunately, they have a much reduced life expectancy. And specifically, I look at modeling this disease and look at the physical properties with a technique called atomic force microscopy.
Okay, that sounds very cool. I don't know how you will do it, but sounds amazing.
Thank you.
You're welcome.
Yeah, so thank you for your explanation. Now we were wondering how is living in Barcelona.
Well, Barcelona is amazing.
Do you like it? I mean,
I like Barcelona.
Okay,
I like Barcelona a lot. What can I say about Barcelona?
I don't know.
So I came here actually to do my master thesis 2 years ago.
Okay. I mean, did you know the city before?
Not at all. I came here because I had friends from my Erasmus, actually. So I came to visit them. And I like the lifestyle. I always wanted to live in the south of Europe. So I said, oh no, I can't say that. I said I will stay here. and I'm still here. Well, I went back, but now I'm back again. And I think it's an amazing city. It's not so big. I mean, it's a lot of people.
It's very big, yeah. I mean,
for me, so the places I lived are London and Berlin, which are super widespread. But for me, Barcelona, I can go anywhere in 30 minutes. Mm-hmm. I think it's beautiful. The people are super friendly. The culture is great. The food is amazing. And the weather for me is really nice. So yes, for this reason, I live in Barcelona. I don't know.
So you're happy here and motivated.
I'm happy. I'm motivated.
That's very good. I mean, I think that we can close now the podcast, the little short podcast. I'm sorry.
But maybe I have a question for you next week.
No, I think that's okay.
We don't have time.
No, we don't have time.
But that's for the next podcast.
Yes, for the next.
Tune in.
Yeah, sure. Tune in.
Okay, Rohit. So you are now a PhD student in an experimental lab, and you work with different types of cells, and also your colleagues from the lab also works with other types of cells. At the same time, I know that you are a fan of this TV series called Avatar.
Oh, yeah.
That in this series, there is like these 4 elements: fire, air, earth, and water.
Yes.
And you can assign like different people with different, how are they with one of these elements.
Yes.
I was wondering if you can also assign different cell types with these 4 elements.
Oh, wow. That's a very, very, very well thought question. And if I have to think instantly, so the first element is the element fire, which is like energy, and its fire spreads very fast. So it's like very contagious. The cell line that comes to my mind first hearing fire element is HeLa, because it's a cancerous cell line, and it's a very aggressive cancer and it spreads so fast. So I would say HeLa would be fire element. Coming to the opposite is the water. Water is known to be being calm and always together. And the cells which comes to my mind are endothelial cells, because these cells they like to be always together, and even they also form these structures like your blood vessels, which also carry these body fluids, which is also like an water element. So like they are holding the water, being a water element. Then we come to earth. Earth is something which is very grounding, very cementing, one of the foundational thing which is required to build or even break things. So, and even to repair. So in that sense, I will say the cells such as fibroblasts, which are like the cementing factory who release a lot of these cements to make these tissues very strong. And they can be also complemented with the bone cells from the bones, like osteoblast, because which forms the skeleton of our body. And for the final element, which is air, which is very dynamic, omnipresent, the essence of life, I would say stem cells would be a really good fit for the element of air, of being versatile. They form the whole organism from the beginning to the end, and they still are present in our body, always doing the work of making us alive as well. So that would be.
Well, that's a very interesting answer, Rohit, indeed, and a very interesting question on as well. So I have another question for you. So PhD is a very challenging task. Conflicts are very possible to arise. Frustration is also a very common thing. And what I wanted to ask you is, according to your experience in academic research, what is the biggest difficulty you have faced so far, and how would you advise younger colleagues that are dealing with similar issues?
That's a very deep question. Actually, it takes me back to my beginning of my university, where I was like starting my bachelor's project, and I was working in a lab, and there, like, we don't used to like talk our research out of the lab, so like there was no out of lab communication. So I found it a bit difficult to try to solve all the problems on my own, even though we were a small group, but it was a very difficult task. I faced the similar issue when I was in my master's. I always longed to, talk about my research and have some brainstorming ideas just with other people from the different lab to have a different perspective. And this bottleneck had been with me almost till my master's. And when I started my PhD after coming to Barcelona, starting here, I found being in a group of lab with two different lab groups being together, it was like a big shock for me in a surprising way that I was able to talk my research out, and people were equally eager to listen to what I do and offer help or advices with their knowledge and with their patience to listen. So, and even we used to have these lab meetings where one guy or one person is presenting in front of 40, 50 people, and you are being shot by a lot of ideas, troubleshooting. So a lot of things are being well taken care of if you are around a group of people who are very, reflective, in the sense that they also feel that, okay, this person is struggling through and I have a knowledge and I will share it.
Mm-hmm. And also common experiences, so they know how
exactly
you feel.
how I feel. I think that's one of the very fundamental point. But also apart from it, I have been talking to people from outside the lab of different, they work on different research areas, and surprisingly, the cell line that I'm working with right now, I found from a group who was working with the blood-brain barrier. So like, this is how connections lead to, cool projects or cool ideas. So to the younger generation, I will say go out of your comfort zone, talk to people, talk out not only your science, but also listen to others, because it's equally important to then being heard to be heard as well. So yeah, that would be my advice. Talk, collaborate, and explore and go out of your comfort zone all the time.
Mm-hmm.
Yeah.
I see. Thank you very much, Rohit, for your answers and for your time.
And thank you so much, Emmanuel and Ona, for this very dynamic and very deep questions as well.
Thank you, Rohit.
Thank you.
So hello everyone, I'm Timothée, and I will interview Petra for his PhD, but I will let her present herself. And I will be with
Alice. Hi everyone. So I'll be asking Petra questions about her hobbies and life in general.
And hello, I'm Petra, and I will be interviewed today about my PhD and my general life.
Okay, perfect. So first of all, Petra, can you introduce yourselves a little bit, your life and you?
Well, I'm from Italy, and I came here to Barcelona first for my master thesis at IBEC, and then now I'm starting a PhD here, and we'll see how it goes, but it will be some fun years. I cannot wait to see how it goes.
Okay, so I'll be asking a question about the project then. Okay, so can you tell us a bit what is your project about and which lab you work into? Also?
Yes, so I work in the Biosensors for Bioengineering lab led by Javier Ramon. And we work mainly with organ-on-chip devices and sensors. So my PhD in particular will combine the two technologies, and the aim is to develop a model of Duchenne dystrophy, in particular the metabolic dysfunction and mitochondrial dysfunctions, and then couple it with biosensors. In particular an SPR one, and investigate the metabolic implications in this disease.
Cool.
Yeah.
That's a great project, quite interesting, and I mean, I think you will discover so many things, and it will be really useful for plenty of people. That's crazy.
We'll see. I hope so, at least.
Okay, so beyond the PhD, what do you like to do in your free time?
Well, I'm pretty an active person. I love running, so you will see me running through the streets of Barcelona all the time. So I get out of the lab, I change, and I go running. So that I really like, but also I really like to explore. I'm down for any new things. Like you just propose a plan, I'm down. I mean, I say yes. So in the weekends also exploring a bit the city, go for some good coffee and meet some friends. And yeah, in general, I like spend time outside in nature with some good people, and I'm happy. Yeah.
Great. Sounds like a nice plan.
Yeah.
For your free time.
You seems to need to run like every time. And that's you think you will running every time during your PhD or things?
Yeah, I think so. I think I will need to, release a bit of stress, because PhD can be really stressful so running, I think it helps a lot. When you're a bit upset for something, maybe I will start bringing my shoes to the lab. Then just change. And when I feel that I'm overwhelmed, I just change shoes, do a quick run, and I'm back for the next experiment. So yeah.
Great. Then it sounds very useful actually.
Yeah.
So it's October. We're starting the new year. You said that you're starting your PhD now.
Yes.
How did you prepare mentally for this new start? I guess you just finished your master.
Yes, exactly.
Masters, and there was the summer in between, so maybe you did something to prepare yourself for this new adventure.
Actually, yes, I finished in at the end of July, so I had a full month, even two, of rest, which I enjoyed. I went traveling a lot. I've been to Indonesia, so yeah, I've seen cultures and stuff. I've been a bit to the beach, so I fully recharge for the next chapter. Yeah.
Great. That's needed.
Yeah. You say that you did your master thesis in the same group that you're doing your PhD right now.
Yes.
That was the choice of the group you choose for the PhD. That's important for you, the mood, the feelings with other people's. It has an impact in your choice, you think?
Yeah, of course. I mean, as I said, a PhD could be really stressful. So if you have a good environment around, I think it helps a lot. Also, in particular, in my situation, I choose to pursue a PhD. Also because I felt really well in the lab. All the people are super nice, super helpful and inspiring. So I guess, I think that in our lab, there's a good atmosphere. So everybody's super nice, and I cannot wait to spend some more time with these people, meet new people, and I think it will be great. Well,
and maybe it's too early to ask this question, but maybe you already have, I don't know, your mind set on something. I just wanted to ask if you have like a dream, like for your career. Like, do you know what you want to do after the PhD or even beyond?
Yes.
Do you have a dream? Okay.
Tough question, yes. But right now, I don't know if I will pursue the academic path. So I think I will move to the industry, some company or stuff like that, and like exploit the knowledge I gained outside and bring it to the, let's say, like to the practical world to improve also all the work in the companies or stuff like that. I don't know in which field I will be fall in, but yeah, for now it's that. I don't know yet. Like, as I said, in which field, but I'm open to everything. I'm super curious. So if you want to hire me, please call me in 4 years.
Great.
Yes, we will put your phone number at the end of the podcast, we think. So I think it's time. So thank you, Petra, and hope you will enjoy your PhD for the 4 years.
Thank you.
Now you're trapped, you don't have the choice.
Well, you will be with me, so we are both trapped. We will survive in some way.
Thank you, everyone, for listening each others, and have fun with other groups.
Yeah. Enjoy science. Bye.
Bye.
So, good morning, Susanna.
Hello.
It's a pleasure to have you here in this podcast today.
Thanks for joining us. Yeah.
You are from IBEC, correct?
Yeah, I work at IBEC. And what is your job at IBEC?
I'm lab manager from a group that is working on biomaterials for neural regeneration.
And
sounds interesting.
So yeah, sounds interesting. But what does this mean? So what does a lab manager actually do?
It depends on the group that you are working in, but in my case, I would say that I managing like logistic and staff, materials, meetings, organizing people, but I'm also doing research in the group. I like to call it like I'm an octopus. So I have one hand in every project helping people.
That's very stressful. Sounds very stressful.
Can be sometimes, yeah, can be.
How do you deal with stress in your workplace, or maybe even when you are outside? How do you stress out your mind?
Normally I would like to do like physical activities like sports, or in my case I daily do weights, and I really enjoy to start new activities. For example, now that I'm from Madrid and IBEC is in Barcelona, I joined to a Colla. That is a group of people that they make like human castles. And I don't know, like trying new things is something that really keeps your mind out of this kind of stress from work.
What level of the tower do you... Are you at the top, or are you at the bottom, or somewhere in between, I guess?
Now I'm in the bottom because it's like the basic, but people is pushing me to start being like climbing once to the second or third level, which is something exciting.
So do you think there's anything in that you're being a foundational support to this tower of people? Would you say there's a parallel to your foundational role in the lab?
That's really good point.
Maybe this is the roles you enjoy doing in your life. I don't know. This is getting a bit philosophical, though.
Yeah, no, it is, but it's good. Yeah, it's true. Like, I would say that I never thought of this, but it's so related. No, it's really...
Oh, nice. And so tell us a little bit about the research that your lab does. Something about neural regeneration? Or something?
Yeah.
So for someone who doesn't know what this is, could you explain it?
Yeah, we specifically work on spinal cord injury caused by impact, like car crash, or a really huge fall, so they get paralyzed. Mm-hmm. So we work on developing new materials, biomaterials, to help these neurons grow and cover this damaged area to reestablish the connection in this spinal cord, so we make this person or people re-able to walk again.
Yeah, sounds super interesting and useful.
And you mentioned you are so speaking about something else, there is not work related.
Yeah.
Do you mention that you are from Madrid? How was your integration in Catalonia?
By my side, I would say that is excellent. Like, I really enjoy meeting new people. And I would say that Catalan people is really nice people.
I mean, the castle you are just get yourself in is very Catalan.
Yeah,
you are getting to the culture. That's very nice.
I would say that in my inside part, maybe I have like half Catalan nationality.
You will have one.
Yeah.
Yeah, that's it.
Buenas tardes, somos Ana y Edu, que vamos a entrevistar a Marc.
A Marc, sí.
Buenas tardes.
Buenas tardes, encantado de estar aquí.
Marc trabaja con organoides en el campo de la neurociencia,
sí,
enfocado al Alzheimer. Teníamos algunas preguntas acerca de cuán difícil es trabajar con estos organoides.
Sí. Claro, pues estos organoides realmente es complicado porque tú los generas a partir de células pluripotentes, ¿no? Que son células madre, células que se pueden convertir en muchos tipos de células. En mi caso, pues los convierto en neuronas, ¿no? Que son las células que se ven principalmente afectadas en el Alzheimer. Y pues estas células se generamos unas bolas y estas bolas generan estas neuronas que tienen una estructura similar a la que observamos en el cerebro.
Uh-huh. ¿Y cuánto te lleva generar estos organoides?
Claro, al final es un proceso muy largo porque puede empezar empezamos con el cultivo de las células y cuando empezamos a convertirlas a neuronas y hacemos la bolita, pues eso, yo creo que al cabo de un par de semanas, 3 semanas, ya podemos considerar que sería un organoide porque ya empieza a expresar cosas que expresan las neuronas y nosotros los mantenemos hasta 4 meses. Unos 4 meses, tenemos algunos un poquito más largos, entonces es largo y tedioso y hay que planificar muy bien cuando empiezas, cuando acabas, porque claro, estos no descansan y si tienes vacaciones, pues te las comes con patatas.
Y entonces, si ya es tan tedioso, ¿cómo compatibilizas tu vida? ¿Es fácil compatibilizar tu vida social y fuera del laboratorio?
Pues la verdad es que
trabajo?
A veces es complicado, pero por ejemplo, yo los fines de semana, si tengo que ir a cambiar el medio el domingo, pues claro, pues si tengo que salir fuera, voy el domingo, pero voy por la tarde-noche. Voy, por ejemplo, al laboratorio a las 8 o 9, si he ido fuera de Barcelona, pues he ido fuera de Barcelona y vuelvo, y sobre las 8 o 9 ya voy al laboratorio. Mira, es un poquito tedioso, pero bueno, es lo que hay, pero me lo logro montar.
¿Posible que te ha afectado tu vida antes y después de organoides?
No, yo creo que no.
¿Os organizáis?
Yo creo que no, yo creo que me organizo bien y que sí que es verdad que tenemos que planificar las vacaciones, porque si queremos mantener organoides vivos durante el verano, pues tenemos que planificarlas, pero sí, yo creo que no, nos apañamos, la clave es la organización.
¿Y qué nos ofrecen estos organoides con respecto a los modelos que hay establecidos ahora mismo?
Pues claro, los modelos que hay establecidos ahora mismo son principalmente modelos de ratones. Entonces los ratones, ellos no generan Alzheimer, no generan la patología ya de por sí, no tienen este tipo de enfermedad, ¿no? Entonces nosotros la introducimos de una manera artificial, de alguna manera, para que expresen la proteína humana y que expresen una especie de enfermedad similar al Alzheimer. Pero ningún ratón recapitula toda la complejidad del Alzheimer, porque un ratón no tiene el cerebro que tiene un humano y un ratón pues no envejece lo que envejece un humano. Entonces es problemático y por eso las células con las que nosotros trabajamos son células humanas. Entonces esos organoides no solo te permiten pues eso, tener unas bolas que son muy manipulables, estudiar mucho tipo de cosas con mucha facilidad y que además éticamente hablando es mucho más ético que no trabajar con ratones, sino que tenemos también el beneficio de que son células humanas y son neuronas humanas.
Al humano.
Exactamente, es decir, tienen realmente un correlato mucho más bueno que los ratones, pero bueno, no son organismos vivos, entonces sí que es verdad que claro, son un modelo in vitro al final, hay que también ser consciente de las limitaciones.
Ok, y entonces, si quieres replicar el Alzheimer en esos organoides, o sea, ¿sientes más interés por el Alzheimer en tema social o más por la investigación? O sea, ¿investigas porque te gusta investigar o de verdad tienes un interés por el Alzheimer
Yo creo que todos los científicos tenemos un poquito las dos vertientes, ¿no? Quiero decir, todo el mundo ha conocido a alguien que esté afectado por el Alzheimer y a todo el mundo le ha afectado de una manera u otra, ¿no? Entonces es una cosa que siempre cuando hablas del tema pues te puede tocar mucho y que realmente es un tema que necesita pues eso, que se avance, que se investigue, y que se puedan encontrar soluciones, que actualmente pues ya se está avanzando mucho y realmente están saliendo soluciones que pueden salir bien, ¿no? Que pueden un poquito tratar o tratar de prevenir, más que ahora mismo las que existen son tratar de prevenir. Pero los científicos también tenemos esta vertiente curiosa, quiero decir, no somos médicos, entonces no tenemos esta vocación tanto de ayudar a las personas directamente. Sí que evidentemente nos interesa ayudar a la sociedad, porque, hostia, a quien no le gusta, ¿no? Pero sí que un científico tiene que tener esta vertiente curiosa. Sin esta vertiente curiosa, un científico, pues, le falta algo, ¿no? Entonces yo creo que las dos son correctas. Es decir,
ya está.
Sí.
Bueno,
pues muchas gracias, Marc.
Marc.
Buenas tardes.
Nos vemos en otra sesión.
Vale, muchas gracias.
Good afternoon. Welcome to the Pipette podcast. Today with us is me, José Huguet, Aga, weird name to pronounce, and Oscar España. Today we're interviewing Oscar España, who's here with us today. Welcome, Oscar. How are you?
Fine, thank you.
Okay. So, you work with CAR T cells, I've heard. Tell us, what exactly are they, and what exactly do you do with them?
Yeah, so nice question. So I would say, first of all, that CAR T cells are one very specific type of therapy, specifically immune therapy for tumors. CAR T cells, what we do is that we take a part of the cells of the immune system of patients and we engineer them and modify them genetically for target the tumor that the patient has. Usually these therapies are now the gold standard for leukemias, so blood cancers, but they are not that good for solid tumors, like, for example, a breast cancer. And so there's a lot of efforts and research on boosting this type of immune therapy for solid tumors. And this is part of my project at IBEC, where we use a very specific technique of spatial proteomics and imaging for doing screenings of different types of modifications that we can add to these CAR T cells for targeting solid tumors. And I'm working on that, on making very whole variety of different modifications to these CAR Ts. And maybe, hopefully, in some years, we can have a CAR T therapy for solid tumors.
That's very specific. Thank you very much for the explanation. But life is not all about the professional work. So what do you do in your free time?
Oh, yeah, I really like that question. So, yes, science, it's not everything. And in fact, one of the things that I do almost every day, it's dancing, not just by my personal happiness, but also as like kind of more professionally in an academia. I dance popping, that it's a very specific type of urban dance. Maybe not a lot of people know popping, and you can look for it.
It's very specific. Like, can you explain us more? What is this about?
Yeah, popping is old school style, which maybe people will be more familiar with hip hop, and it is derived from that. It is true that it's very niche, but I just love it. It's a way for me for, like, after focusing the whole day working with CAR Ts, I go dancing and I can, like, relax. And yeah, that's quite what I do.
It's only popping, or is there any styles that it's your favorite?
At the moment, it's mainly popping, but I've been dancing other types and other styles, from ballet, like is very classical, to other more commercial styles, urban dance. But yeah, like my passion, it is popping.
It's a huge palette of different styles. That's very nice.
So what else do you do apart from that? Like, obviously you told us you do some other sports and that, so if you care to elaborate quickly in under 1 minute.
Yeah, of course. So I've been quite sporty during my life. When I was younger, I already danced, but I've been doing other types of sports, mainly judo for like 6 or 7 years in my life, and then also rugby for around 9 years. It is quite a change passing from judo, rugby to dancing, but I like them all, and I'm thinking to return to do a bit more of them as I used to do, but also PhD takes lots of time. So I'm thinking about it.
Well, that's been all for today in the Pipette podcast. Thank you for listening and tune in to the next episode.
Thank you very much, Oscar. It was a pleasure to host you and get to know you better.
Okay, good morning to all. Maria, so nice to have you here today with us. It's nice to be here.
Thank you, Maria, for coming to us. And it's a pleasure for us to be here interviewing you. And next, please, if you can introduce yourself to know about you.
Okay, I'm Maria Escola. I am from a small town in the south of Catalonia. And I came to Barcelona to study my bachelor's in biochemistry and my master's in microbiology. And next month, I will be starting my PhD in biotech.
That's very exciting.
Yes, cool. And can you explain why do you choose the combination between chemistry and microbiology?
So I started my bachelor's in biochemistry, and doing my bachelor's thesis, I was working with plants, mostly doing genomic DNA plants, and I was also doing clonings. And I found out that working with bacteria is way more easier than work with plants, because a bacteria culture takes one day, like one night, to just grow, and plant cultures take like a month to grow the plant, and then also cell lines are really complicated to work. So I found that I wanted to have an easier lab life working with bacteria.
I feel like that was a good choice for those that we are coming on the weekend and doing all the not fun stuff with human cells. I think that was a good choice.
Yes.
And for me, I would like to ask you something more about your personal life, maybe. So how do you envision your perfect Saturday, for example?
So on Saturdays, on weekends, sometimes I go home to my small town, and my dad cooks pancakes in the morning, and I like to have those pancakes in my garden while I am reading a book. I mostly like crime novels, and while my dog is playing in the garden. That will be my perfect Saturday morning.
Sounds like the dream. I want to join you sometime now.
Of course.
Les cèl·lules, que són les unitats més petites vives del nostre cos, tenen la informació més important a dintre del nucli. Però aquesta informació està com protegida per el nucli, per la membrana aquesta nuclear. Llavors, com s'activen els gens de què tant hem sentit a parlar, que després faran que les cèl·lules es puguin moure, es puguin reproduir, etc etc. Doncs això és una mica el que estudia l'Aina. A veure, Aina, explica'ns una mica. Què vol dir això de que mires com s'activen, com fan les molècules per arribar fins al nucli?
Doncs resulta que aquestes cèl·lules que coneixem estan totes plenes de molècules. I cada molècula, com si diguéssim, s'encarrega d'activar un gen en concret. Llavors, els gens, com ha dit la Janet, estan en el nucli. I aquestes molècules típicament estan a fora en el citoplasma. Llavors, quan s'ha d'activar un gen en concret, la seva molècula ha d'entrar al nucli. Però, òbviament, això no és un procés fàcil. El nucli té unes portes super sofisticades que són les que diuen quines molècules poden passar i quines no. Llavors, quan una molècula ha d'activar el seu gen, el que fa és... necessita energia de la cèl·lula i doncs aconsegueix passar per aquesta porta tan sofisticada gràcies a altres molècules que llavors s'enganxen a ella i la fan passar.
Llavors tu has d'intentar veure això tan i tan petit, a veure com passa per aquest porus o aquesta porta nuclear, no?
Sí. Llavors jo el que tinc són unes molècules que jo afegeixo artificialment a dintre d'aquestes cèl·lules, que són molècules que es poden comprar però s'assemblen molt a les que hi ha a les cèl·lules, i estan marcades fluorescentment, o sigui, emeten fluorescència. Llavors és veritat que són molt petites, llavors si n'hi haguessin moltes i tot estigués ple d'aquestes molècules, nosaltres veuríem inclús al microscopi un continu, no podríem distingir-les, però si en poses molt poques, llavors veus puntets brillants. I quan tens puntets brillants individuals, sí que pots veure molt millor i predir molt millor d'on venen i on són. I és així com puc veure-les des que estan a fora fins que passen per dintre d'aquestes portes al nucli, fins que ja finalment són a dintre del nucli.
Uau, això que dius és molt maco, sembla que dediques molta feina en intentar estudiar això, i jo imagino que quan arribes a casa tens ganes de desconnectar i deixes ja de mirar aquests detalls tan petits, d'intentar buscar coses. Què fas tu fora del laboratori?
Doncs, allò que diuen, no?, que quan tens un problema molt gros, si en tens un altre encara més gros, t'oblides del primer. Llavors jo el que faig és que me'n vaig a fer curses d'orientació al bosc. Llavors, jo això és una cosa que no l'he feta des de petita, o sigui, jo no tinc desenvolupat el sistema orientatiu en el meu cervell gaire bé. Llavors jo em poso al bosc amb un mapa, amb una brújula, i intento anar a buscar totes les fites que diu el meu mapa. Sovint, què passa? Que jo no trobo les fites ni a la primera, ni a la segona, ni a la tercera.
Encara que les marquis en fluorescència, tampoc les trobes en aquest cas?
Mira que estan allò fluorescent taronja, sí. Però de vegades inclús a dintre del bosc jo t'asseguro que no les veig. Llavors, doncs mira, està a dintre del bosc perduda, intentant buscar la fita número 49, en aquell moment, no, és el problema més gran en aquell moment de la teva vida i és fantàstic que el teu problema més gran sigui estar perduda al mig del bosc buscant la fita 49.
Sí, és perfecte. Així és com es perd la gent. Molt bé, doncs molt interessant. Fins aquí acabem el nostre small podcast. Esperem que us hagi agradat i per més informació cliqueu al banner.
Adeu.
Hola. Estamos con Natalia Castro, Armando Cortés y yo soy Ángela Ramírez. El día de hoy vamos a entrevistar a Armando, que nos va a hablar un poco de su doctorado. Así que sigue, Natalia.
Armando, cuéntanos un poco qué es lo que te inspiró al mundo de la ciencia.
Bueno, yo cuando pienso en la ciencia siempre recuerdo mucho a mi tío, porque, bueno, él es un ingeniero civil, pero es muy apasionado de la ciencia en general. Y cuando yo era pequeño siempre me hablaba de animales, me hablaba de matemáticas, de todas las cosas que tienen que ver con ciencia. Entonces, desde ahí creo que desarrollé un gusto muy fuerte por la ciencia. Entonces escogí una carrera que juntaba muchas cosas. Soy ingeniero en biotecnología. Entonces incluye todo lo que tiene que ver con ingeniería, pero también entender a los sistemas vivos y todo eso. Entonces, pues sí, de ahí viene el gusto por la ciencia y pues de ahí viene también el interés por hacer un PhD.
¿Y cómo es que llegaste aquí a hacer el doctorado en el IBEC?
Yo estaba en la universidad y tenía una profesora que tenía un colega y él formaba parte del grupo donde estoy investigando ahora mismo. Es el grupo del doctor Javier Ramón. Y él estaba investigando sobre biosensores y esto. Así fue como conocí a Javi, entonces nos entrevistamos y pues él tenía un proyecto para mí. Y así es como yo llegué.
¿Y de qué va este proyecto? ¿En qué se enfoca tu investigación?
El proyecto es sobre dos enfermedades que a nivel clínico se observa que muchos pacientes comparten. Una es el hígado graso y otra es la sarcopenia. Entonces lo que hacemos es explorar la relación que tienen estas dos condiciones en pacientes que están enfermos del hígado. Esto lo hacemos con modelos de enfermedad. Es decir, pueden ser ratones, pueden ser cultivos in vitro y nuestro approach es utilizar modelos in vitro. No utilizamos animales y también usamos la tecnología de Organs on a Chip.
Me parece espectacular tu proyecto, sobre todo esa parte que no usan animales y bueno, creo que va a llegar a tener un gran impacto. Esperemos que sí, te deseo todo lo mejor y Me gustaría preguntarte, cuando llegas a casa, ¿qué es lo que haces? ¿Qué es lo primero que haces?
Pues lo primero que hago es saludar a mi perro porque usualmente está ahí en cuanto abro la puerta, está esperándome y está muy contento y después llega con un juguete y lo empieza a morder y espera que juegue con él.
Entonces tienes un perro, ¿cómo se llama?
Sí, se llama Comisario.
Oh, genial. Wow.
Sí, es un perro que adopté en México porque él se metía a nuestras clases en la universidad y entonces estaba dentro del salón de clases con nosotros. Le estábamos buscando casa, pero pues eventualmente ya no se fue. Me lo quedé yo y después salió la oportunidad del PhD, así que lo traje para acá.
Ok, qué interesante. Entonces, ¿te gusta mucho estar con tu perro, jugar con tu perro?
Sí, a mí me encantan los perros. En realidad es algo que descubrí hasta que adopté a Comisario, que me gustaban mucho los perros y también se me daba bien comunicarme con ellos y eso. Entonces, bueno, de hecho, me certifiqué ya como entrenador canino.
Wow.
Y es como mi side hustle, mi hobby y sí, mi segunda pasión. O a veces creo que es la primera. Qué feliz.
Bueno, pero me parece también importante ver ese lado humano detrás del lado científico de todos nosotros.
Sí, al final todos tenemos no sé, tenemos muchos gustos. No me creo que haya un solo científico que solo se interesa por la ciencia. Entonces es interesante también cultivar otros hobbies, otros intereses.
Bueno, pues creo que cerramos aquí la entrevista y un gusto
Muchas gracias.
entrevistarte, Armando.
Bye.
Chao.
Hola, buenos días a todos. Estamos aquí en Raticos de Ciencia. Soy Alba Calonge
y Ainhoa Ferret.
Y estamos aquí con Alex Jiménez, que va a empezar ahora su doctorado en dinámica de tejidos. Y bueno, te queríamos preguntar, Alex, para empezar, como que expliques un poco qué estudias más específicamente.
Hola, gracias por invitarme. Yo voy a empezar a estudiar los ritmos circadianos de las células. Estos ritmos circadianos vienen a ser como una especie de manera que tienen las células para poder medir el tiempo, entre muchas comillas. Básicamente se trata de unas variaciones que hay en concentraciones de ciertas proteínas que a su vez inhiben la expresión de las mismas proteínas, de forma que se crean unos ciclos más o menos con una periodicidad constante de 24 horas que las células utilizan para poder saber en qué momento es más óptimo a realizar distintas funciones como la división celular, por ejemplo.
Qué interesante, Alex. Muchas gracias. Aparte de tu vida como científico, ¿sabrías decirnos alguna curiosidad más sobre ti?
Bueno, pues a mí siempre me ha gustado mucho la música. Desde pequeño empecé a tocar el piano y luego el violín y ahora mismo soy músico amateur, no es nada profesional, pero la música se ha convertido en algo muy importante entre mi vida y me sigue gustando muchísimo tocar y pues eso, no sé, para mí es muy importante.
Pues muchas gracias por acompañarnos en el día de hoy. Alex, un saludo a todos y nos vemos en el próximo episodio.
Come to our post. We are with a PhD. We are here with a PhD student from IBEC. And welcome. And what's your name?
Hi, my name is Gergő, and I'm from Hungary.
Wow, that is amazing. And I want to ask you about why do you come here to Barcelona to research, and how do you feel about it?
So I came to Barcelona because I got invited back by my current PI after my master's project. I did my master's in Glasgow, Scotland, after moving there from Hungary. And during COVID, I needed a master's project to complete. I got in touch with several places, and this is where I got the most appealing offer. And after a little detour of searching for jobs, I ended up coming back to the PhD. And so far, so good. I really enjoy my work that I do here. I do think with moving around for so many places, I finally am happy that I am sort of stationary in a spot where I can find so many good people and interesting people. It's a very international community we have in IBEC and in Barcelona both. So yes, obviously all this moving comes with its challenges, but I'm doing fine.
Perfect. Thank you for sharing.
Okay, so you talk us privately before a little bit about your research. It's really interesting what you do in translational research, specifically in the field of cancer detection. So this is a field that has a lot of patients, has a lot of money. Still, many things are not solved and could be improved, and it's what you're working on. But also there are still some limitations that could be economically. Could you explain us a little bit more what you're aiming at? What could be the potential beneficials, the limitations, timelines?
Of course. So in our group, we focus on real-time cancer metabolism and its detection with a technique called hyperpolarized magnetic resonance spectroscopic imaging, which sounds very fancy. But in reality, we are using an MRI to, instead of images, looking at real-time metabolism. MRI mainly focuses on hydrogen nuclei. In our case, we focus on carbon-13. And we use a technique called hyperpolarization, in which we enhance the baseline signal sort of thing of a substrate. We introduce that to our cancer cells or organoids or whatever we're trying to look at, animals, for example, and within a time frame of approximately 30 seconds to 1.5 minutes, we can detect metabolic products. And the main challenge of this field right now is that it's a very small field. In every conference, you meet with the same people from all around the globe. It is a very expensive technique. Preclinical MRI that we have in the lab is approximately 1 million euros for servicing. A clinical polarizer is about 2 million euros, and to get all the chemical probes you need for it and all the cryogens to cool down the magnets to superconductive temperatures, it is ending up to be a really expensive technique. Obviously, if this was something we can implement in the clinics, it would be up to date, one of the best cancer screening techniques, because it's non-invasive and that is on its own a very big advantage. But unfortunately, most MRIs around the world, they are in hospitals. You cannot just go to your local CAP and ask for an MRI scan. And also, clinicians are not yet convinced because the technique is still in clinical trials. A lot of people, especially on the engineering side, try to make this a more accessible technique. But I think we still need quite a few years until we can just go to a local CAP.
We can wait.
Yeah.
Well, thank you very much for this short talk.
Of course.
See you later. See you in the next.
Thank you for your questions, guys.
All right, so
Hi, good morning, Asia.
Morning,
hi.
Hi all.
I'm Luisa, and here we also have Leon. Yeah.
Hi.
Hi, Leon. Hi. So, Asia, today, are you going to talk about your experience here in Barcelona?
Yes. Should I start like?
No, don't worry. Okay. Just tell us a little bit about your life in Barcelona.
Oh, we're gonna let it. So. Okay. How long have you been in Barcelona for? I'm here since 1 year and a half now. Yes, it will be 2 years in January. So. Okay.
Oh wow!
Yeah. Already 2 years here. Yeah. Yes, I'm from Milan. And did you start with the PhD? Like, did you come here for the PhD, or were you here? No, actually, I started with an Erasmus post-graduation. So 6 months of Erasmus, then 1 year of research assistant. And I'm starting the PhD right now in September.
Cool. And your PhD, what's going to be about?
I'm a medicinal chemistry, and I'm working on a project in which I have to synthesize derivatives of dextroamphetamine.
Whoa, so, yeah. You're going to be Breaking Bad.
Yeah. Totally. But then there are some other projects, like one of epilepsy, that's a neuropathic pain that I'm working on. So, yeah, more than one project, but so exciting, I would say, yeah.
So you like brains, huh?
Yes, I like awakened brains. Yeah.
You are awake brain.
Yeah,
Oh,
that's
how
my...
cool.
And how did you become interested in brains?
Yeah, it was a joke, but
No jokes here.
no, it was not my choice. I mean, it was a project that we had in the group, and it was very interesting, the chemical part and the computational part. So then our group is expert on neuron and brains in general. So let's work on it.
Good. Thank you, Asia, for your great job.
And apart from the PhD, what else do you do in Barcelona?
Okay, I joined in a group of batucada, so percussion, yes.
Yeah.
And I love it, actually. And I go climbing with my friends. I practice yoga. What else? I enjoy Barcelona, the seaside, the mountains. Also with the interviewer, Luisa.
Oh, yeah.
That is actually my colleague. Oh, my God. Oh, my God.
Oh, but that's a spoiler, Asia. We're not
sorry,
supposed
but
to read this.
I'm sorry. I just realized that
then
you are you. This Luisa.
Okay. Then I need to challenge you, huh? With some scientific questions.
I'm ready.
Are you ready? So,
I can...
what's your plan after synthesizing this molecule? Are you going to test them in somehow?
We already test.
Oh, but you personally, did you learn some?
I only watched and do it also in zebrafish larva.
Oh, nice.
Yeah, once I did once. But then they tested also in rats, and then there I only see one experiment.
How was it? Was
tiring, because they stay like 4 hours without sit down, without going to the bathroom, without drink, and and they with a mask. So, but it is they work like. In this condition every day or every time they do an experiment. So, but at the end, it was a good job. I mean,
thank you, Asia. Thank you very much. That really sounds like you got to broaden your skills and knowledge.
Yeah. So
thank you for sharing.
Thank you very much for
thank you guys for inviting me.
And always.
Hello, Ainhoa.
Hello.
Today we have here in the podcast Ainhoa Gonzalez, which is a PhD student from IBEC. Thank you, Ainhoa, for coming.
Thank you.
She's working in new therapies for bladder cancer. So we wanted to know, what are you doing, which kind of therapy, why this is beneficial for the bladder cancer?
Yeah. So we use nanoparticles, which are really, really tiny particles, and we load into them something that makes and modifies the genes from cancer cells in order to kill them. So yeah, we are working on that. It's really, really...
How is it going? Depending on the day, no?
Yeah, depending on the day. We have like frustration days, but also good days. So we are working on this.
And when you finish your PhD, do you have plans or expectations after your PhD?
So I'm finishing my second year, and now I don't really know what I'm gonna do. Because like having a PhD is nice. It opens like a lot of doors, but also if you want to stay in academia, the salaries are really, really low. And for the preparation that you have, I don't think that the salary adjusts.
It's not fair.
Yeah. So I'm thinking what to do next. Yeah.
Well, you have plenty of time, no?
Yeah, 2 years to think about it.
Thank you, Ainhoa. So we will interview you in 2 years.
Thank you.
Hasta aquí, Raticos de Ciencia. Te esperamos en el próximo podcast.